Okay, here we're going to look at the MHC proteins, which stands for Major HistoCompatibility proteins. Put the note here of know thyself. This is how cells are able to interact and recognize one another. These MHC proteins are very important. So class 1 and a class 2, and we'll look at those in more detail. So starting with that know thyself here, quote, know the enemy and know yourself. In 100 battles, you will never be defeated. So this is part of the immune system. It needs to be able to identify the enemy, but it also needs to know itself and be able to recognize what self cells are so it doesn't attack those. So the first step in identifying the enemy. So what's our first kind of process here? Well, we have to recognize our own cells, and every cell in the body carries surface markers. Those surface markers are called MHC proteins. Specifically, they're called Major HistoCompatibility proteins. And these MHC proteins are different for each individual, and they act as self-markers. So class 1 has basically one binding site here to the cell, and class 2 has 2 binding sites. We can see an example here of our class 2s and class 1s. So these are class 2s, these are class 1s, these are 1s, and these are 2s. This indicates even further which ones came from a maternal line, which ones came from a maternal line and a paternal line, from mom and from dad. So identifying the enemy is number 1. So we have antigen presenting cells, sometimes abbreviated APC cells. They ingest foreign particles or partially digest them. In this case here we have a macrophage encapsulated this bacterium in a vacuole. They then combine pieces of foreign particles with MHC proteins and move them to the surface. So our lysosomes here, our digestive enzymes, are fusing with this bacterium, causing it to break down. And then here's our antigen, and these are MHC class 2 receptors, move the digested bacterium and presenting it on the cell surface. T cell receptors can only interact with cells that have a combination of MHC and antigen. So this is why it's important to have these MHC proteins incorporated in that. That's helping identify as a self and not destroying the entire cell here. This image here we're going to spend a little time on is the antigen processing and presenting steps. So here we notice we have a pathogen. In this case it's being ingested here. And this pathogen or extracellular antigen is indicated here, 2 different pathways are being taken in by the cell, being engulfed by the cell. Phagocytized would be the proper term to use. And this antigen-presenting cell, which is a dendritic cell, is placed into a vesicle. So we see both of these different pathways of antigen being acquired from either inside the dendritic cell here, or required from outside the environment here, being placed into a vacuole. These ingested pathogens are digested by the digestive enzymes in the lysosome, and they're interacting with the antigens. Now this lysosome is releasing these enzymes. These enzymes are breaking this bacterium down, maintaining the antigen from the digestive pathogen. These antigens bind with MHC proteins that enter the vesicle here. So we notice it's occurring with the bacterium, and also in the case here where we have our extracellular antigen going through that same process. It's important that these are able to bind with the MHC class 2 protein so that they can be presented on the cell. As we progress further, our MHC proteins are now carrying the antigens and are released from the vesicle and travel to the outer surface of the cell membrane. If they never reach the outer surface, our T cells wouldn't be able to bind with them, meaning if our MHC proteins bound with the antigens and were stayed inside the cell, there'd be no point. Our T cells can only bind with receptors on the cell surface. So it's very important this dendritic cell is now presenting antigens on the cell surface so that it's able to activate T cells that bind with the MHC proteins. This is helping the immune system be able to recognize these foreign invaders or these antigens. We require these antigen processing cells, APC cells, to go through this process to then display these on the surface with the appropriate MHC protein so our T cells can recognize this as self and recognize this as a foreign antigen and then initiate a potential immune response. So hopefully going through and understanding this very complex process was a little bit easier. You're welcome to pause the video and take a look at this image here so you can better understand the antigen processing and presenting process.